Welcome to Shri Chhatrapati Academy Pune Study Ayush Forum for the students by the students. Right? Hello to all the future doctors of this independent nation. Welcome to the field of treatment. Our today's agenda is human physiology modern from modern perspective only, right? So, before starting today's lecture, I want to give you some motivational thought. So, that thought is education is the key that unlocks the golden door to the freedom, which means when you are well educated, you are well freedom, you will get well freedom, you will have nice knowledge. So, to take your own decisions, you will not be dependent on anybody. So, education is very important, which is given by Sir George Washington Carver, right? Today's our topic is modern topic from hemopoietic system, which is plasma proteins. In the previous session, we have studied about blood, right, and body fluids. So, plasma proteins. Plasma proteins are the important proteins of our body. Proteins are needed, right? So, there are three types of plasma proteins mainly, which are albumin, globulin, and fibrinogen. These two are called as serum albumin or serum globulin because they are only present in the serum. We know what is serum? Plasma minus fibrinogen, right? So, this is plasma minus fibrinogen. So, when fibrinogen is not present in serum, these two are present. So, it is usually known as serum albumin or serum globulin. These are their normal levels. Serum albumin, serum globulin. Total protein percentage is 7.3 gram per deciliter. Deciliter means 10 to the power minus 1. Okay. Which is the average of this two value. Okay, their molecular weights are given. This is also a memory part. If you want to become a good physician, remember this. Now A to G ratio, that is albumin to globulin is to ratio, which is very clinically significant. You can observe it in your lab, etc. So when you observe it in the lab or laboratory report of any person, there is A is to G ratio, albumin, globulin count. Why it is? So this is the ratio of albumin to globulin plasma proteins in the plasma called as albumin globulin ratio. Important indicates some of the diseases of liver and kidney. Normal Ag value is 2:1. This is the value. Now what happens is in the some diseases means regarding the liver, for example, let us take as hepatitis, means hepatitis. There is a swelling or there is some damage in the liver cells. Hepatocytes are liver cells, right? So there is a one point that plasma proteins are synthesized in the liver. So when liver do not synthesize this albumin ratio decreases. And globulin is also synthesized in blood with response to immunity. So what happens is this albumin ratio decreases and globulin ratio increases. So as normal value is 2:1, when it becomes 1:2, there is some pathology. It is a clinically having some problem, right? Same with kidney. When albumin is less or globulin is more, there might be some pathology. We can consider it. Origin of plasma proteins. In embryonic stage, plasma proteins are synthesized by mesenchyme cells. This mesenchyme, what is a mesenchyme? Mesenchyme is a vital undifferentiated embryonic tissue layer surrounding the yolk sac. I will tell you about this when we will study about human embryology, right? In adult, it is synthesized by reticuloendothelial cells of the liver, which are reticuloendothelial cells, it is a system which is a mechanism for, it is made, it is a sometimes it is a system, right? And in that system these cells are called as Kupffer cells, which are phagocytostic cells, which help in immunity, which when they see some when there is some foreign particle, this cell goes and attacks them. This whole system of the body is called as reticuloendothelial cells, and that cell, different organs has different cells. Liver has Kupffer cells, lungs has dust cells, also by spleen, bone marrow, disintegrating blood cells, etc. These are also synthesized by spleen, because there is a what happens is RBC disintegrates and all the parts, hemoglobin is formed, and when this globin part is separated, heme part is separated, globin is a protein we get at, okay. Bone marrow, disintegrating blood cells, etc. Gamma globulin is synthesized by B lymphocytes. B lymphocytes is also help in immunity. So B lymphocytes secrete gamma globulin. Functions of plasma protein: helps in coagulation of the blood, because fibrinogen is essential for clotting factors. Okay. Defense mechanism: gamma globulin plays a major role by acting as antibodies. So these proteins are also called as immunoglobulins, right? Very simple. Transport. Now this is very important point, mark it. Albumin and alpha and beta globulin are important for transport of hormones and enzymes. These are vectors for transportation of hormones and enzymes. And gamma and beta globulin also play important role in transport of metals in the body. For example, when there is needed of iron in the body, iron, right? So that iron is absorbed from intestine and transported for formation or wherever it is needed with the help of this alpha and beta globulins. These are vectors. Role in maintaining osmotic pressure. Osmotic pressure. What is osmosis? Osmosis is movement of water from its higher concentration to lower concentration, right? At capillary level, most of materials are exchanged between blood and tissues, right? Blood and tissue at a capillary, this is arteries, these are capillaries, and these are venules. So these are capillaries. Here there is so much exchange between tissues and vessels, right? However, because of their large size, plasma proteins cannot go outside of the capillary. These plasma proteins exert oncotic pressure because they are not going outside, they are stucking here, they are blocking the way. So there is a actually when their action happens, there is an increase in the pressure in the blood vessel. This pressure is nothing but the osmotic pressure, sorry. Albumin exerts 70% of oncotic pressure by plasma proteins, which is 25 mm of Hg, which is it is a unit of pressure, you know that. Albumin is more in blood as with the, so now see what happens is albumin, then globulin, and then fibrinogen. This is the level. Albumin is high, then the order is of globulin, then the order is of fibrinogen. So as albumin is more, it will exert more osmotic pressure. Nothing. Simple. Right? Properties. Now oncotic pressure. Plasma proteins exert on oncotic pressure and osmotic pressure. Okay. Value is 25 mm of Hg. Very important role is of albumin because it is more. Lion's share, we can say. There is a lion's share of albumin in oncotic pressure. Specific gravity means relative density, which compared with water it is 1.0.26. Now buffer action. Buffer. What is the buffer? Buffer is the entity which means that whenever there is any pH change, it resists, it avoid that pH change. So as we know that blood is pH is 7.4, which is basic in nature. So buffer action happens by the acceptance of H plus ions. Now how? Now in our previous classes we have studied that proteins. Now plasma are the proteins. We have studied the structure of proteins. Protein structure is, there is a carbon, then there is a NH2 group, then is a COOH group, then there is a H group, and there is a R variable group. This is variable in variable proteins, right? But we have seen that in our body these proteins are not present in this format. These are present in Zwitter ionic form, which can exchange. Zwitter ionic form is present. There is a difference in the charge. Okay, now what happens is, when there is a action of when there is a basic buffer, there is a acceptance of H plus ions. Now what is basic? Amino acids at high pH. High pH means there will be less H plus. So here there will be not H plus. So here there will be only single O minus. So what happens is, when this when there is an increase in the H plus ions due to some anyhow condition, this H plus binds with this O minus, and hence the pH is maintained. Same condition occurs when there is a low pH. There is an excess of H plus. This H plus, excess of H plus. Then what happens? Then here is H plus. Then this H plus, what happens? It comes here and attaches here. NH ammonium, NH4. Sorry. NH3 only happens, and here there is a carbon atom. So this H plus attaches here. This action happens in the blood when wherever there is a high pH, and this happens wherever there is a low pH and high H plus ions. Now there is one hypothesis which is given by Sir Starling. So its name is given as Sir Starling hypothesis. What is hypothesis? This is only the statement, it is not proven, right? So hypothesis: osmotic pressure plays an important role in exchange of blood and cells through capillary membrane, right? There is a plasma proteins, this is capillaries, these are veins, these are arteries, these are veins. So here there are capillaries, right? Now osmotic, now the hypothesis is the net filtration through the capillary membrane. Now the filtration due to this, here there is a kidney, this is a glomerulus of kidney. Net filtration through this capillaries is directly proportional to hydrostatic pressure, means the pressure of the blood of this is stable format. Static means stable format, minus oncotic pressure. Oncotic pressure means the pressure exerted by plasma proteins, right? This is the hypothesis, no clinically proven, but at with respect to the logic, it is correct. Now it helps in role in acid base balance. Now only we have seen that it maintains buffer action, it maintains ionic balance in the body. So albumin plays an important role in acid base balance because it is present in more amount. This is due to buffering action. We have seen responsible for 15% of total buffering action of the body. Role in viscosity: plasma proteins provide viscosity because they are protein in nature. Okay, and important to maintain the blood pressure. So blood pressure, when there is a solid substance, fluid moves very fast, so pressure will be more. So it also helps in maintaining the blood pressure. Albumin provides maximum viscosity than any other protein because they are present more amount. Role in erythrocyte sedimentation, which is PCV. Globulin and fibrogen are important for Rolex formation. What is a Rolex? Rolex is a thing which happens in the red blood cells. Now imagine these are the red blood cells. This attachment of red blood cells in pile coil like format, pile of coins format, this is called as a Rolex formation. You can consider it as there is a granum situated in the chloroplast of the plants. They are also attached like this. So imagine in that way. So this is Rolex. So important plasma protein in the formation of Rolex is globulin and fibrinogen, and they are responsible for erythrocyte sedimentation rate, which is important diagnostic and pre-diagnostic tool, right? Now, role in suspension stability of RBC. During the circulation of RBC, they are very uniformly suspended in the blood. This is, imagine this is as veins, and there are red blood cells. They are very uniformly given, not here it only, here it is, or here it is, which will obstruct, which will make here Rolex formation, there will be clot. So it's not like this, uniformly distributed. This property is called as suspension stability of the RBC. The plasma protein which help in this phenomenon is fibrinogen and globulin, right? If their level increases, so it will be for here so much Rolex formation, so there will be clot and there will be many pathological condition related to the heart. Role in production of tryptophan substances. What is tryptophan? They are today, in nowadays, they are called as growth factors. They are essential for growth activities. Tryptophan substances are necessary for nourishment of the tissue cells in culture, that is cell growth. These substances are particularly produced by leukocytes from plasma proteins. They are nothing but similar as that of cytokines and lymphokines, which are the growth factors. Role as reserve proteins. They are the last source of energy. What do you think the last source of energy? Whenever there will be fasting condition or inadequate protein supply to our body, then these plasma proteins breaks by the tissue macrophages in the amino acids and distributed again to the body to fulfill the protein needs, right? Now there is one experiment which is called as plasmapheresis. Now earlier called as Whipple experiment, because it was given by Sir George Whipple, right? Used to demonstrate the importance of plasma proteins. We will see the conclusion further. I will tell you first the experiment. What is the thing is, you have to take an animal dog. You have to separate, you have to remove the blood from the dog's body, and you have to separate the plasma proteins from it, and then wash with saline and again reinsert to the dog. What happens is the dog goes under the state of certain shock. So after this, when plasma proteins were given to the dog, within the 7 days the dog came into the normal state. So what this experiment tells us is, conclusion, important plasma proteins importance for survival of human body, and synthesis of plasma protein is done in the liver. So this experiment was performed. So thank you for today. We will meet in our next lecture. Till that, study well and ask doubts. More the doubts, more the